oxidized carbohydrate. If fermentation does occur, there will be a bubble in the upside down tube. They call it, there's two names for it. They call it an inverted tube, and they also call it a Durham tube sometimes. So I would know that.
Yeah.
So if there's a bubble in it, that's a yes for fermentation. Because when fermentation occurs, gas is released, and the indicator will also change color. It'll change, it starts out red, and if fermentation occurs, it will be yellow. So
We will need to know the colors.
yeah, so be aware of the color. Let me think. And the reason that it turns yellow is because it's acidic. All you need to know is that, well, let me see. Okay, that's
Why is it important to do these tests?
it is important to do these tests. Oh, did you want them to answer?
No one?
No. Does anybody have an answer for that? It's just basically a way to like differentiate between different bacteria. Okay.
Why is that important? So he's going to ask you clinically how to use this. Why is it important? Why would you care if a bacteria can ferment? It's so you can know your enemy. So you can know how to destroy
So you can
it.
identify the bacteria.
Exactly.
Basically.
You have to know what it does before you can say, okay, well, this is how we kill it.
Right. Yeah, and in more scientific terms, bacteria are often identified by their enzymes, which can be detected by observing bacteria's ability to grow on specific compounds. So for instance, in this test, can this bacteria grow on carbohydrates? So if yes, it will be yellow, and there will be a bubble, because acid is produced and gas is produced.
So it's an invisible bubble?
Yeah, you'll be able to see it just with your eyes.
What does it do to the bacteria if it becomes more acidic? It denaturates it. This will be on another test.
That would be like the lecture.
Not the lab test. My bad.
Okay. So the second test that you guys are going to look at today, and this one, for me, this one is really hard to wrap my head around for some reason. I'll put it back here. Okay. The MRVP test. And this one is actually like two separate tests almost, because first you add methyl red to the broth, so that's what the MR stands for. Methyl red. Okay. And the pH is below 4.4. So it's acidic. The color of the tube, the color of the culture will be red. Okay. This is after you add the methyl red. If neutral products are produced, in this case, I would know, because I think he might have even asked this on one of our quiz. Make sure you read through exercise 14, okay? Because acetoin that is the neutral product. It's a neutral product. Then it will just stay, it'll stay yellow. So that's the first step of the MR test, okay? Step 1. Step 2 is the VP, which kind of is a complicated name, but I don't think you need to remember it, but it's Vogue's Pro-Scour test. And basically, in this one, you add, okay, you're going to add two things. You're going to add VP reagent 1. Sorry, it's really bad handwriting. And then VP reagent 2. And then you have to wait like 20 minutes before you check it again. And a positive VP test, there will be like a little pinkish, the whole thing won't turn pink, but just like at the top of the broth will be like pinkish. Pinkish. Okay, and a negative VP test, if it doesn't change, I think it just stays, either stays yellow or turns, okay, it turns light brown, so. So negative VP will be light brown. So this one's the most lengthy test because you have to wait. Like I think in your book it says like 15 to 30 minutes before you look at it. And there is one more thing that you might need to know. This little chart right here kind of shows you certain bacteria. It will turn red for a period of time, and it might turn back to yellow. I forgot to mention on the carbohydrate fermentation, it'll be red if the byproduct is ammonia. Then it will be the pH, let's see, pH will be basic. Because ammonia raises the pH, it actually makes it basic. Okay, so I hope that kind of, again, you're going to look at it in lab today, and so it'll probably be easier when you actually have your hands on and you're actually doing the tests. Okay, and the last test in exercise 14, which this one is pretty straightforward, is the citrate test. Oops. So, just what you're looking for in this one is whether the bacteria has the enzyme citrate lyase to break down citrate to form pyruvate. So again, I would definitely read through and make sure you understand, because he might ask you, you know, in the citrate test, bacteria with enzyme citrate lyase breaks down citrate to form blank. Like you might have to write in pyruvate or something like that. So just be aware of that. This one's pretty easy. Either it stays green or it turns blue. So if it turns blue, it means the bacteria broke down the citrate. And the byproduct of that is ammonia. See, ammonia is kind of, it's like, yeah, and it'll stink. But it's a byproduct in a lot of these tests. So just remember ammonia, it causes the pH to rise and it makes it alkaline or basic. And I guess it's interchangeable. So the indicator turns from green to blue when it is alkalized. So that indicates a positive citrate test. So that one's a little bit easier than the other two. Okay, so know what color each test begins and what color it turns depending on whether it's a positive or negative test, and know why it changes color, okay? And it usually depends on the pH because of the byproducts produced.